So to put this lesson in context, we are on the health and disease topic, number 5 for paper 1, our final topic for paper 1. The T shows the topics that are only going to be in the triple award paper, and monoclonal antibodies and the core practical will be discussed once you return. But for today, we're going to focus on a lesson that comes attached with a bit about antibiotics, which is all about drug trials. Now, this has certainly been relevant with the COVID vaccination, hasn't it? I'm sure you've been paying attention to the news, but the release in the press about the announcement of the Pfizer vaccination and now more recently the AstraZeneca Oxford vaccination has been a huge relief and a huge way forward as we fight this battle against COVID. But not only has this been the quickest vaccination to get around, but it's happened because scientists have worked 24 hours a day in order to be able to not only come up with a solution, but also to go through a vigorous route of trialing and testing the drug to work out what is the dosage, what is the effect of it, what are the side effects of it, so that once it goes out to the public, as it is now, that scientists can confidently say that it is safe to do so. So your lesson heading is drug trials, and we're going to be thinking about how new vaccinations and drugs are developed. And what I would like you to do, to get into context why it can go so horribly wrong if we don't go through a strenuous process, I want you to watch this fascinating video. It's about 4 minutes long. It's about a drug that wasn't properly tested, and you will see the awful consequences of it. So if you pause the video now and have a look at the YouTube clip in your list. Isn't that just incredible? And in fact, the COVID vaccinations have not yet been tested on pregnant women or women who are breastfeeding. So they become one part of the population that are unlikely to get the vaccination anytime soon. And it's the lessons that we've learned from thalidomide that helps make us very, very cautious. But well done to that amazing female scientist for standing her ground and doing the right thing. So what do we need to do to produce new medicines? And what stages do we go through before it actually goes through to patients? That's what we are going to look at. Now we know that medicines come from everywhere. We know we've learned recently that from the bark of the willow tree, early tribes figured out that if they had a headache, that chewing on the bark of the willow tree, the headache disappeared, and that's where they realized that inside there is a chemical which we now know as aspirin. And we've now been able to synthetically make that chemical so that we don't have a headache anymore. Scientists do a lot of computer modeling. They do a lot of studies in the lab. As you've known, if you've watched, there's a fantastic program that you should be able to find on iPlayer on the BBC about, it's a panorama program about the AstraZeneca Oxford vaccination, and it ties in so beautifully with this. I would really recommend you take the time to have a look at it. Okay, so in order to develop new drugs, there are lots of stages that we need to go through, and so I want you to stop now and have a look at the second video in your list of instructions, and I want you to particularly focus on the stages that scientists go through in order to test that new drugs and new vaccines are in fact safe to go out to the public. And you're going to be using this to create your activity that's coming up next. So pause the video. Okay, so using what you've just learned from the video, over here on the left-hand side, you've got step one of a couple of stages of testing. And these are all mixed up. So what I would like you to do is read through them carefully. And then in your book, you're going to write them out in order. So first, put them in the correct order. But I would also like you to add a reason why that stage is important. Okay, so you need to find the first step and then the correct reason for it. And you need to write this in your book under the heading Drug Trials. Once you have done, you can take out your purple pen and you can then purple pen using the next part of the video. Okay, so here we have the answers. The preclinical stage of testing is done on cells or tissues in the lab to find out whether the medicine gets into diseased cells and has the desired effect. So we wanted to get into those parts of the body that it needs to treat. Number two, testing may be carried out on animals to find out how the medicine affects the body systems without risking human health. Now, of course, there's lots of ethical issues to do with this, and sometimes some animal parks have some monkeys that have perhaps been kept in a lab for a long time, and it's really, really sad to see. And we need to weigh up, you know, is it better to test it on animal? And if something is wrong, therefore not tested on humans. It is a very tricky and ethical question that we have to ask ourselves. Step 3, a small clinical trial is carried out on a few healthy people to find out whether the medicine is safe for humans to take and that harmful side effects are limited. So that's why they do it on a very, very small amount of people first. So for the Oxford vaccination, I think it was a couple of hundred people or maybe even 1,000 people that they tested the vaccination on first. And then number 4, they then do, once they're happy with that, they take it to a large clinical trial without as many people as possible. And actually what they want is they want a broad range of age groups, of races, of ethnicity. And in fact, with the Oxford vaccination, they made sure it was even tested in different countries as well. What they want to do at this point is they want to figure out what is the right amount of medicine, the right dose, and whether different people have got different side effects. And what was very interesting is that with the Oxford one, there was a little bit of a mistake, which actually made them realize that there was a better dose to use than they had initially thought of. Right, so now those are the first couple of steps. Let's have a look at how these clinical trials are carried out. So all these points must be copied in your book, and you might want to just pause and underline some key words. It's going to be important that you can recall the different steps. Okay, now let's have a look. Over here, I've got 3 bits all jumbled up. From the video, you should be able to work out what the order it is and then match up the reasons as you did before. So pause the video, have a go, and then once you've done, we are going to purple pen our answers. Okay, how did you do? Right, so step number 5, patients are randomly put into 2 groups. And by the way, the random part is really important, so there's no bias at all in the testing. So they're randomly put into 2 groups. One group is given the drug and the other is given the placebo. And you need to know that the placebo, it's like a sugar pill. It doesn't have the active ingredient of the drug on it, in it, and that is to ensure that the effect of the drug is due to the drug itself rather than any psychological effect that the patient might have because they think they're now being treated. Number 6, Clinical trials are blind. Now, what we mean by that is that the patient doesn't know whether they are being given the placebo or the real drug. And the reason for that really is so that the effect is down to the drug rather than any placebo effect as well. And the last point is about the double-blind trial. Now, it's double because not only does the patient not know whether they are receiving the actual drug, but the medical team don't know either. And this is so that the doctors monitoring and analyzing the results aren't in any way influenced by their knowledge. So it's not that they're giving that patient extra treatment because they think that they are getting the real drug. The results are really going to be a revelation to everybody, the patients and the medical team. And this takes out the bias from the process, and that's a very, very important step. Okay, so what I'd like you to do now is just stop for a moment and see if you were asked a 6-mark question on describe how a new vaccination could be tested. I would like you to see if you can recall all 7 of these steps and maybe even push yourself to give the reasons as well. So just test yourself and then check it against your notes now clearly in your book. Okay, right. Now, as a final step, here are a couple of questions for you to answer. I want to see how many of these can you answer. You can just randomly write down the numbers, maybe in the shape that it is, write down your answers, and then you can pause the video, and then check the answers, maybe rewind if you need to. Right, so how did you go testing yourself? Were there any a little bit tricky? Remember, this is not a test, so you can always go back to your notes and use your book to find the answers. But here are the answers, and I hope that you got pretty much all of them. This one at the bottom, it builds memory cells, resulting in a faster and better response. Remember, the concentration of the antibodies is so much more increased during the second vaccination. Okay, and one final thing, you've worked really, really hard. One final thing is the Scrabble gremlin in me. Here we have a number of letters. I can make a 14 point word by using pathogen. What I'd like you to do is see how many words you could score related to the whole health and disease topic. Write them down in your page. Write down the number of points you get for it. And let's see how many of you can get above the 14 points. This is a great exercise to remind yourself of the array of scientific terms that you need to know really well to get the best grades in your GCSE biology. Fantastic work. Well done. Next lesson, we are going to do a quiz. So if you have any time left over today, what I can strongly suggest you do is start to put together your revision notes on this topic. Soon after we get back, as we have pretty much finished the topic, we will be doing an end of topic assessment, and I want you to be well prepared for that. Well done, and keep safe. Don't forget to subscribe or like the video. Thank you ever so much.